Welcome to the latest Novogene webinar, Introduction to RNA-seq Analysis, Content and Pipeline. I'm John Cafferkey, Marketing Manager here at Novogene Europe. This is the fifth in our lockdown webinar series. We'll be sending you details of the rest of the program after the webinar. A bit of housekeeping before we begin. The webinar will last 1 hour with a 10-minute question and answer session at the end. Please submit your questions via the question and answer chat functionality you'll see in the bottom of your screen. We'll answer as many questions as we can. If we can't answer your questions live, we will get back to you by email after the webinar. I would like to introduce you to your host for today, Dr. Seamus Stack. After graduating with a degree in biochemistry from University College Cork in 2005, Seamus completed a research master's degree at the National University of Ireland, Galway, in molecular oncology under the tutelage of Professor Kieran Morrison. After a 2-year period working as a molecular biologist in the antivirals department at Pfizer's Global Research and Development site in Sandwich, Kent, Seamus undertook a PhD in virology at the Centre of Virus Research at the University of Glasgow under the Professor Avind Patel. Following the successful completion of his PhD, Seamus then worked as a senior research and development scientist at Mars Group, an independent manufacturer of molecular diagnostics based on Merseyside. He worked there for 6 years, where his focus was on development and manufacturing of isothermal amplification kits against various targets. He joined Novogene in October 2019 and is regional account manager for the North West and West Midlands region of the UK. Seamus, over to you.
Thanks very much, John, for the very kind introduction, and welcome everyone to our webinar. Sorry, I'm just trying to move. Sorry, can I able to move to our next slide? Sorry, John.
Is it not playing ball, Seamus?
It's not playing ball at the moment, sorry.
I would unshare and reshare it.
Sorry, excuse me. And screen. Sorry, excuse me. Okay.
Technology is always there to trip you up.
Sorry, pardon me, everyone. I have to stop share and share screen. Excuse me. Okay. Let me try this. So, yes, sorry. Can everyone hear me okay? Yeah, sorry, hi. Thanks, John, for the kind introduction. My name is Seamus Stack. I'm the Regional Account Manager for Novogene in the United Kingdom, covering the North West and West Midlands area. I'd like to give you, welcome to the webinar, and I'd like to give you a quick introduction to the company. So Novogene is a leading provider of genomic services and solutions. We were founded in 2011, and our mission is to improve life science research, clinical diagnostics, and human healthcare. We're the largest sequencing center in the world. We have the sequencing capacity to sequence over 280,000 human whole genome sequences per year. And we have a range of platforms and equipment to conduct this sequencing. For short read sequencing, we have the NovaSeq 6000 and HiSeq X Illumina platforms, whereas for long read sequencing, we have the Sequel, the PacBio Sequel platforms and the Nanopore PromethION platforms. To store and manage the large levels of data that we produce daily, we have a supercomputing and data center, which allows effective management of customer data. So Novogene is a rapidly growing company and a young company. It was founded in 2011 in Beijing, and we have sequencing labs in Tianjin and Nanjing in China, but we also have sites in the United States, in Singapore, and we have a sequencing center in Europe, in Cambridge. We're also hoping to, in the near future, open a site in the Netherlands. So for our European customers, they would ship their samples to our site in Cambridge, and depending on the service that the customer requires, sequencing would occur there in Cambridge, or we would transship the samples to our sites in China, and we do this constantly in a safe, speedy manner. So customers' samples are always well looked after. In terms of our service portfolio, we have a large service portfolio that covers almost all aspects of next generation sequencing. This slide highlights the key services that we provide in terms of, if you're interested in human genome sequencing, we do whole genome sequencing, whole exome sequencing, and target capture sequencing. If you're interested in epigenetics, we do whole genome bisulfate sequencing, chip sequencing, and RIP sequencing. If you work on microbial sequencing, we have a lot of experience in amplicon sequencing and shotgun metagenomics for microbiome analysis. We also do de novo sequencing and re-sequencing of microbial genomes. If you're interested in plant and animal studies, we do de novo genome sequencing and whole genome sequencing, as well as reduced representation genome sequencing. If your work is on transcriptome or RNA related sequencing, our core services include mRNA sequencing, non-coding RNA sequencing, single cell RNA sequencing, and ISO sequencing for full-length transcripts. This is the focus of our webinar today, and my colleague will go through these services in more depth shortly. So to why, let's say, you should choose Novogene as your service provider for your genomic needs. We have, let's say, 6 core key elements of our service that are that stand apart. First among these is our quality, in terms of the quality of our data, which actually surpasses Illumina's Q30 guarantees, but also and as well as our customer service. Our capacity, our range of sequencing platforms allows us to take projects of size, whether large or small. In terms of the size, does not matter. Our technology, the range of platforms that we have that allows flexibility for customers to choose to refine the project needs that they require. Our turnaround time is very fast. This is due to our large sequencing capacity and the range of platforms we have, as well as the experience and expertise of our staff. Our bioinformatical analysis is unsurpassed. 40% of our staff are educated bioinformaticians, and we can provide publication-ready data for customers and their requirements. We are an innovative company. We have over 157 software copyrights and 27 patents, and we're constantly looking to adapt and evolve. And it is these characteristics that have allowed us that have given us customers in over 60 countries, let's say, representing over 4,000 universities and research institutes. Our data contributes to publications in Nature, Cell, Science, and other high impact journals. And indeed, we have a section of our website which demonstrates these publications and our role in their success. So this slide is to introduce you to our excellent and really friendly sales team throughout Europe. We cover different regions of the continent. And if you have any queries and you need to get in touch, we'll be delighted to help you. To contact us, you send us an email at europe@novogene.com, and then we will assign you to your local regional account manager, and they will be in touch with you and support you throughout your project to help you with all your customer needs. So with that, I will introduce our webinar speaker today, Dr. Wei Li. So Dr. Wei, we got our bachelor degree from Nanjing Agricultural University, majoring in life science and technology, before moving to the United States, where she gained master's and PhD degrees from Clemson University in South Carolina, working primarily on cotton transcriptome analysis. She started her career at Novogene Europe as a senior bioinformatics engineer, but has been promoted to a senior product manager, where she is now in charge of service development at Novogene Europe. So with that, I will hand over to you, Wei, for the webinar.
Thank you for the introduction, Seamus. Can you hear me well?
Yeah.
Okay, now I'm going to share my screen. Can you all see my screen?
All good, Wei, all good.
Okay, sounds great. So good morning, everyone. Thank you for joining us for the webinar, and I'm Wei. Today, I will give you a brief introduction about the RNA-seq analysis content and pipeline provided by Novogene. So in this presentation, we will cover 3 parts. First, we will go over the major services we are providing right now for the RNA-seq. And the second part, we will use the Eukaryotic messenger RNA-seq pipeline as a demo or as an example to show you how we perform the analysis. And in the last part, we'll introduce you some cases to give you a brief idea about how our service can support your research. As shown on the in the top figure, so we can see different type of RNAs in the eukaryotes can interact with each other or compete with each other. So for the eukaryote study, we have different services to target each type of RNA. For the coding RNA or the messenger RNA, we will sequence them on either NGS platforms such as Illumina, or we can do the long-read sequencing using the PacBio platform. And to perform the analysis, you don't have a reference available for this because we already have a de novo pipeline ready. But for the non-coding RNAs, such as long non-coding RNAs, circular RNAs, or small RNAs, we have different services for each of them. Or we will have the network study by using the whole transcriptome sequencing service. But for those analyses, we will only collect the data from the NGS platforms. And we do need a reference genome available to run the analysis. For the prokaryotic RNA-seq, if you are interested in a single species, we can provide the prokaryotic RNA-seq as they have different structure in their genome or they have different regulatory mechanism for the transcription. So the pipeline for analyzing the prokaryotic RNA-seq will be a little bit different than the eukaryotic ones. If you are interested in the microbial community, we will also have the meta-transcriptome service available. In this one, we will focus on the active species showing up in this community. So we'll perform the species annotation and to study their abundance as well by using the meta-transcriptome. For each project in Novogene, we will follow this workflow. So you can either send us the total RNA if you extracted them already, or we can provide the extraction service for you as well. And after we receive the sample, we'll perform the sample quality check, and then move on to the library construction, library quality check, sequencing, and analysis. If you have a very special library or you prefer to prepare the library by yourself, we can also accept the pre-made libraries. For all of the projects, if you don't need the bioinformatic analysis from Novogene, we will release both raw data and a data QC report to you. So from this report, you will check how your data look like. For the projects with bioinformatic analysis, in addition to the raw data, we will also release an analysis report, which covers all the introduction of each step and some demo results from your project. And we will also include an analysis result package. In this one, all the figures and tables will be publication ready, so you can use them freely for each of the publication. Now let's move to the second part, and we will go through the eukaryotic messenger RNA-seq pipeline in details for each step. So when we do the RNA-seq, like we want to study some human genes or human gene expression, actually for all of our body cells, they all come from the duplication of a same cell or same stem cell, which is the fertilized cell. But why some of the cells will have different functions? Like the cells in our eyeball, they can see different things. But for the cells on our skin, they will only protect us from the atmosphere or some environments. The main reason, or one of the main reasons, will be the gene expression, because they are expressing different RNAs. And in all of our, or majority of RNA-seq analysis, we're trying to address two different questions. The first one, we want to identify what genes has been expressed or what kind of transcripts are there in my transcriptome. So I term it as a transcript annotation. So we want to, what are they? And the second question we want to answer is the quantification. We want to how many of each transcript are there. Maybe the cells will, actually for all of the cells will have the same genetic DNA. So why, if they are expressing the same gene, why the abundance are different in my different type of cells? So those two questions will be the major two objectives we want to address in our analysis. And here I'm showing the messenger RNA sequencing, messenger RNA-seq pipeline for the eukaryotics. And we always start from the raw data from the very top. And then we check the data quality, and only the clean data will be used for the future steps if you request the bioinformatic analysis with us. And basically in this pipeline, we also trying to answer the two questions I proposed in the previous slide. So the left half is kind of showing the quantification related questions. So based on the gene quantification, we were trying to find out the differentially expressed genes and some functional analysis. While the red half is showing the other question. So what are they? Is there any alternative splicing? If there is any mutation in my transcriptome? So in this part, we will address that question. And some analysis showing in this kind of dashed line square is only for human and mouse. So those two species are very well annotated and studied. We will include more detailed analysis for those two species. So let's go through each step in details. First, we start with the raw data quality check or a raw data QC. So in this step, we will include multiple assessments of your raw data. And here I only show the quality distribution as an example. So we will present the data or base quality distribution in the figure as shown here. Left and right is showing read 1 and read 2 if you ordered pair-end sequencing. And for NGS platform, it's very common that we got lower and lower quality as the read elongates. So this is kind of quite common. But for the Illumina official guarantee, we will use a Q30 as a very important parameter to assess how your data look like. For their official guarantee, they can guarantee Q30 higher than 75%. But for Novogene, we will guarantee Q30 higher than 80%. So basically, this number, the higher the better, and we exceed the Illumina official guarantee for our services. And the other thing we will perform for raw data is to classify your raw rays into different categories, showing on the right. So some of your rays may got very small inserts, and in that rays, we will capture some sequence from your adapters. So we classify those rays as adapter-related rays, shown in green. And some of the rays may have so many uncertainties, such as N's. And we will classify these rays as containing N rays. And some of the rays may have very low quality, so those will be low quality rays. The last category will be the clean rays. So hopefully we'll get as much as possible for the clean rays. For the project without bioinformatic analysis, only your raw data will be released. And at the same time, we will release this data QC report as well as this classification report. So you can check how much of your clean rays is capturing from your sequencing. And for the projects with bioinformatic analysis, we'll perform the data filtering based on this. As I mentioned, only the clean rays will be used for the future steps. So now we have our clean data ready for the following analysis. So let's move on to the next step. The second step will be mapping. So we want to know where those reads are coming from. And we map the short reads from the NGS platform back to our reference genome. So through mapping or through alignment, we are going to use the sequence similarity to confirm if this read is coming from this region of my reference genome. And for the reference genome information, you can either check the reference genome from those 3 listed browsers, or when you fill in your bioinformatic form, we will also include a list of reference genome that you can choose from. And on the right, I just show a figure for a human project to let you check the read distribution or the mapping status along the different chromosomes. As shown on the right, on the outer circle, so each of the region will be labeled as 1 chromosome, and those gray region is kind of showing their density of the mapped arrays. To visualize your mapping results, we will use the IGV software. So in this software, you can load your reference genome on the bottom. So in here, you can see their detailed annotation with which region is my exon, which region is my intron for each gene. And in the middle, you can also load your mapping results, which has been saved in BAM file. So you load those BAM files for different samples, and then you can check the coverage or sequencing depth of your each of the sequencing depth related to each gene. And in this software, you can change the scale very easily. So this one will allow you to check the sequencing coverage at a single base resolution. Through mapping, we can also collect the information for the quantification. So this will be our 3rd step. In this one, once we got the raw counts, so some of the, if we want to compare between different genes within the same sample or genes between different samples, there will be some factors will affect the raw read counts mapped to the same gene, such as for the red gene versus the blue gene, the green gene. So the green gene is definitely longer than the red gene. It's quite possible that we got more reads mapped to this region versus the red region. And if we got very different sequencing depth or data amount from different samples, then the read counts mapped to the sample 2 may be much higher than the sample 1, just because we got different data amount output. So to help us easily visualize or compare the gene expression level between samples or between genes, we're going to normalize our raw counts against those 2 factors. FPKM, fragments per kilobase per million, is a very nice or commonly used normalization method for the RNA-seq. So in this one, it will allow us to normalize our fragments of each gene against the 2 factors. One will be the total read counts, which is also the sequencing depth, and the other will be the gene length. So as shown on the bottom, we normalize fragments counts for each gene against the gene length and the total read counts. So with this, after this normalization, you can easily compare each gene in different samples. So we will present the FPKM distribution in different format by Novogene. So you can use either curve to show the FPKM distribution for different samples, or you can use a box plot or Violin plot to show their distributions as well. So basically, majority of our genes will have very low FPKM value. That totally makes sense because our cells want to use our energy very efficiently with only few portion of our, very little or small portion of our genes being expressed at a specific time. By using the FPKM value or result, we can also perform another 2 analyses. That will be the correlation analysis showing on the left or PCA analysis showing on the right. So on the left, it's kind of showing the similarity between each pair of our samples. So the R square or the correlation coefficient shows the similarity. The higher the number is, the more similar those two samples are. So as we can see here, the left 3 samples are actually 3 replicates from the CK group, and the left 3 samples are actually 3 replicates from the 6-hour group. So by checking the correlation analysis result, we can tell we got very nice replications within each group. And we got a very nice treatment effect between my control and my 6-hour group. So this is very helpful for us to confirm my experiment has been set up properly. So it's very useful for us to check if there's anything going wrong during my sample collection. And on the right is another way or similar way to cluster your samples while using the PCA. So each axis is showing actually one major factor or principal components to affect the data distribution. As we can see here, 3 replicates from the 6-hour and 3 replicates from the CK group actually are separated along this PC1. So it actually makes sense that we got a very nice treatment effect. So this actually helps us to confirm our correlation analysis results. And the other way that you can get, the other information you can get from here is, if you really want all of your samples in the same group clustered together very well, so maybe if you check this axis or this the PC2, you will see the CK1 is actually separating from CK2 and the CK3. So if you really need to pick up an outlier, so this one may be a better choice compared to the other two. So now we can move on to the differential expression analysis part. So here in the differential expression analysis step, we will not use FPKM value for the analysis. We will always use raw read counts as our input for this step. This is because for majority of our analysis software, such as DSeq2 or EdgeR, they are all requesting raw read counts as input because they have their own normalization algorithms in them. So we will follow their own algorithm as well to run the differential expression analysis. And here is just a screenshot of your bioinformatic analysis form. So you can define the treatment group versus the control group. And based on this information, we will give out the distribution of your DEGs showing in bar graph for different comparison groups. And we can also present the Venn diagram to show the unique DEGs or common DEGs shared by multiple groups. So the next two figures I'm going to introduce will be two of very popular presentation method for the RNA-seq result. So the first one will be the Volcano plot. And again, you need to define our comparison groups. As shown here, we use A versus B as an example. So in the Volcano plot, we will plot all genes based on their expression, based on their log-to-fold change and their p-adjusted value. So as we already defined the comparison group A versus B, so from the x-axis is showing the log-to-fold change. That means positive and negative log-to-fold change between A versus B is actually showing upregulation and downregulation respectively. And here p-adjusted equal to 0.05, the dashed line is showing where the significance cutoff is. Anything above this dashed line will be significantly differentially expressed, but below will be no significance. So in this way, as we can see from this figure, by comparing A versus B, we will have around 4,600 significantly upregulated genes, which are shown in red, and around 3,300 of significantly downregulated genes, which are shown in green. So this can give us an overview about how my differentially expressed genes distributed in my whole data set. The second one will be the heat map to present the RNA-seq data. So this is a very commonly used method as well. In this one, we will cluster all differentially expressed genes from all different groups in this heat map. As you can see, from the top half, if the genes are all so red and blue shows upregulations and downregulations respectively. So from the top half, if the genes are all upregulated in my left 6 samples, but downregulated in my right 2 samples, those genes will be clustered accordingly. And on the right here, we can also add more information to help us to track more details of this gene, such as the gene length. You need to refer to this legend or this gene tab. And basically from this legend part, we can tell most of them will be protein coding genes, and also check the chromosome information by using this legend. So why we want to present the clustered differentially expressed genes? Because in some of the analysis or in some of the species, not all of the genes has been well annotated. Maybe some of the genes we have no idea about how they will involved in this biological process. But by clustering them together with similar expression patterns, it will help us to predict their potential function, because they have similar responses behavior if you compare to some genes with well annotation information. So in this heat map, it will help us to study the functional information of some unannotated or weakly annotated genes. So that will be the heat map for the differentially expressed genes. And with that, with our differentially expressed genes identified, we can move on to the next step, which is the enrichment analysis. So we will run Go enrichment, CAG, reactome, DO, or DisGeneNet enrichment analysis for the differentially expressed genes. And we will also run GC, gene set enrichment analysis for all expressed genes. So for each of those databases, they are actually focusing on different aspects of the biological processes. Like the gene ontology, they will give a very universal term to describe the function of this gene, which includes their cellular components, molecular function, or their biological process they will involve in. Well, from the CAG database, they will introduce a lot of biological pathways those genes may involve in. By using different databases, it will help us to discover the significant pathways those differentially expressed genes contribute to. And our result will be quite similar, like we use the bar graph to show some significant enriched terms. So the terms will be listed on the left. Or we'll use the scatter plot, including the significance, the enriched terms, and also the gene numbers associated with this term. So this scatter plot will add more information to the enrichment result presentation. And for the CAG pathway result, there will be a very special point. That will be when we presenting the pathway here, as shown in this figure, we will also label the differentially expressed genes in colors, as shown in green here, to let you know how your differentially expressed genes involved in this CAG pathway. And the last one based on the quantification information will be the PPI, protein-protein interaction. So we will use the string protein interaction database, and we input our gene information. And with that, it will give us or help us predict the potential interactions from this database. And then we can input the results from the database into this software, which is the Cycloscape. And then you can produce this network showing on the right. So each node will showing one protein, and the edge is kind of showing their potential relationships between two proteins or two genes. So in this way, you can, it will help you to check their potential interactions between different genes or different proteins. So basically everything in the previous slides is covering the quantification analysis and all the related analysis based on the quantification results. So the next part, we will introduce some analysis associated with the annotation part. So the first one will be the alternative splicing. In this way, the alternative splicing will introduce a lot of diversity of our transcripts. So maybe one gene can express the tons of different transcripts, because there will be a lot of alternative splicing events occur during the transcription. So here we listed 5 major events here, such as the skip exon or alternative 5' splicing site. And to present the alternative splicing, we will use the sashimi plot. So in this plot, we'll use the arcs to show the proportion of mapped arrays. So if you see thicker arcs, that means more genes has been mapped to this region or mapped to this junction event. And the peak will show the abundance of the arrays mapped to this exon. And from here, if you check the 2 isoforms showing on the bottom and also the abundance showing on the top, we can see in the orange samples, we actually have the more reads mapped to this kind of exon. So that means in the orange samples, they will have more isoform 1 compared to the red samples. And the other structure-related analysis will be the variant calling. So in this one, we focus on 2 types of mutation or variants. First one will be the SNP. It is a single nucleotide polymorphism. And the other one will be the multiple nucleotide mutation, which is including indel, including insertion and deletion, so indel. And in this one, in this analysis, we will include 3 major information. So first, where are they? So where is my mutation showing up? And the second, about their function. Like some of my mutation in the messenger RNA, they will change the amino acid sequence in my protein level. So that will be the missense mutation. And in the last step, we'll also predict how this mutation affects my normal biological process. So we will predict their impact. Maybe some of the mutations showing in the protein level will introduce very severe disease or very abnormal development. So in this one, we will include all those 3 steps in the variant calling for your RNA-seq. And the last analysis we will carry will be the fusion gene study. So here, as shown on the top, a lot of tumors will carry a lot of fusion genes or fusion transcripts. So this one is showing an example from the colon tumor. So these new transcripts are actually fused from 2 separate genes, showing on the top, the left one and the right one. So with a very high level of these new transcripts, it will contribute to the pathogenesis of the colon tumors. And we will also save the results in the BAM file or in your fusion gene analysis. And you can load them or visualize them using the IGV as well. So here, the arrows is actually pointing to where the junctions are. And in our data release, we will include a README, which is an instruction for how you can use your data, our raw data, FASTQ files, and our mapping results. And also we will have our complete report for you to introduce each step and a result package showing on the right. So if you check the result package or folder, you can see each folder is actually corresponding to the analysis step we have gone through in the previous slides. And in the report, we will also include the analysis software list with their software name, their version, the major parameter you want to keep an eye on, and some remarks in our software list table. Except the coding RNA, as we introduced by now, and there will be a lot of non-coding RNAs as well. And for each non-coding RNA, we also have the different services in Novogene. If you want to study only one type of non-coding RNA, so in addition to the annotation and quantification, we will include an extra step, which is a target prediction, because those non-coding RNAs majorly involve in the regulation of the gene expression. So we want to predict their potential targets. And for the whole transcriptome sequencing, we focus on the network study between multiple types of the RNAs. Here, I just listed the microRNA analysis pipeline as a demo. And you can see on the bottom, we include the target prediction and further analysis. So now let's move to our last part, which is the case study. And you will have a brief idea about how our service can contribute or can support your research. So our mission is to advance genetics and to improve life. So our potential application actually can be used for all of those listed areas or fields. And here I listed basically three basic applications for your research. The first one I want to introduce will be to build a transcriptome profile. So this paper is trying to set up a transcriptome profile of rat genes after the spinal cord injury at different stages. They use rat and they perform the injury treatment, and also they include the control group. And at different time points, they either rate their performance based on their movements or responses to different stresses. And they also use the RNA-seq to track their molecular information. So in the RNA-seq analysis, they track different time points, such as the acute or subacute or chronic phases, and to see how the RNA-seq data change at different phases. And to analyze it, we follow the basic pipeline. We check the raw data, we map them, quantify them, and perform the differential expression analysis and enrichment analysis. And in their result, they will have an overview of their sequencing data, like their Q30, their data output, and an overview of the mapping status, like how many reads has been mapped to my reference genome. After quantification, they ran the PCA plot to see how their samples cluster together with each other. And they ran the Volcano plot to present RNA-seq analysis, and also Heatmap and some other cluster analysis to see how their differentially expressed genes cluster together with each other. And our contribution to this project is we build a global gene expression profile at different stages. And we we identified the critical genes and signal pathways involved in these responses. It built up a foundation or a reference for the future related studies. And the author also uploaded the data to the NCBI database. So if you want to rerun the analysis, or if you want to compare the data with other projects, you can feel free to download the data from the NCBI database. And the second case or second application I want to introduce is to study the regulatory mechanisms. Because when you study the non-coding RNAs, you want to find out their potential targets. As shown in this paper, so they want to study the hydrolysis in the nascent hextraploid wheat after the cross. and they include messenger RNA-seq and the small RNA-seq. So their materials will involve a lot of different species, such as the tetraploid female parent, diploid male parent, and their baby generations, and also a common wheat cultivar in China. And they also involve the different tissues, such as seedlings, speck, and seeds. And one of the questions they want to answer is to find out the molecular basis for the hydrolysis. And in our analysis, except the basic one, we also include the target prediction step for the microRNA. As you can see from those small portion of their results, because I cannot include all of them. So they sequenced their messenger RNA and they found a very clean or very nice cluster pattern between samples, between tissue tabs, and the microRNA distribution among different type of tissues within each species. And as if you can see here, so as the microRNA log-to fold change increase, we actually seeing messenger RNA log-to fold change decrease. So it's kind of showing the negatively correlationship between the microRNA expression level and the messenger RNA expression level. So in here, one of the conclusions will be the small RNA is actually involved in the regulation of the hydrolysis in the tetraploidy wheat. And except that, if you only want to focus on one type of the non-coding RNAs, you can just choose different type of non-coding RNA service. But if you want to cover all of them, then we can offer you the whole transcriptome RNA-seq. So in this one, you only need to send us more than 5 microgram total RNA. We will construct 2 libraries from this. One will be ribosome RNA removal directional RNA library for sequencing non-coding RNA, messenger RNA, and circular RNA. The other will be small RNA library for sequencing small RNA. In addition to the basic or standard analysis for each type of the RNA, we will also include a network analysis, including the correlation analysis for each 2 types of RNAs between any 2 types of RNAs, or a co-expression network study among 3 types of RNAs. So there will be 2 more points I want to emphasize here. First, for this circular RNA, it is not linearized RNA removal library for this one. So if you really want to cover all of circular RNAs, we will suggest you to choose this library tab. If you want to focus on the network study, so this library will be enough and is sufficient for you to follow up. And the other thing is, if you want to sequence all of those different types of RNAs separately, you may need to prepare more than 10 microgram of total RNA input. But for our whole transcriptome RNA-seq, we only need half of them. 5 microgram will be totally enough for us to move forward. And here we just show you we can identify different type of RNAs and their network study through the whole transcriptome RNA-seq analysis. And the last application I want to introduce that will be to track the transcriptomic responses. As we can see here, you can actually join different type of analysis, such as whole transcriptome, whole genome methylation study and the RNA-seq study, as the methylation level will affect the RNA-seq expression level as well. So if you want to study how they affect each other or how they interact with each other, we can also perform the joint analysis between RNA-seq and the DNA methylation study. So here we show an example performed on apple trees, and they are studying the diverse flowering capacity. As you can see here, we can collect the methylation profile for them, and we can also construct the RNA-seq or transcriptome profile for the research. And then by comparing the methylation level and their RNA-seq expression level, you can see their relationship or their correlation for different flowering genes. So on the top, we just listed the contribution we can involve from Novogene. And back to this one, Seamus already introduced. So we provide different type of services. So if you are interested in any of them, or you want to join them, you want to combine any multiple of them, you can always contact us. And we have contributed to more than 430 publications with a very high accumulative impact factors. To support either storage, analysis, or sequencing, we have a very super powerful computing system at the end. So you don't need to worry about how you can guarantee the quick turnaround time for your analysis. With that, I'd like to thank all of your attention, and I'm glad to take any questions from here.
Thank you very much, Wei. That was an excellent presentation. Yeah, so in terms of, we do have time for a few questions from the audience. Actually, so I'll begin. Catalina Ramila asks, when you're looking at FPKM distribution on the plots that you showed during your talk, does these represent each gene in the sample or the average FPKM per sample?
So it depends. If you are checking the FPKM in one sample, that will be the FPKM for each gene in one sample. And if you have multiple samples, we will have, and you define a group including multiple samples, and you want to see the FPKM for this group, then that will be an average FPKM for this group, which calculated from multiple samples.
Okay. Samar Elarbani, he asks, can the correlation and PCA analysis be used for single cell data samples? As it's his understanding that there's no FRPM in single cell RNA-seq.
I think they are asking about the 10x single cell RNA-seq. So for the correlation, if I recall it correctly, for the PCA analysis, it is included in the 10x single cell RNA-seq analysis, because from that they can identify the major factor or the major principal components to separate different cell types. So PCA is definitely included in the 10x single cell RNA-seq, but for the correlation analysis, I don't think we have that part in our pipeline right now. And for the single cell RNA-seq analysis, they will perform a little bit differently using different softwares and algorithms to run the differential expression analysis, because they want to find out the most variable markers first, and then they will move forward with those information.
Okay. A question from Nancy Statopoulou. Do you only read the maps onto the IGV browser, or could you also use the UCSC?
So that will be, so for the UCSC, so that is a genome browser, and we can also access the different types of the reference genomes as well. So we have 3 options for you to choose from for the reference genome. Like you can use Assemble, you can use NCBI, or you can use the UCSC. But for the IGV browser, so that is just a browser for you to visualize your data. And I think that browser can also allow you to access to different remote or online reference genome as well. So in my opinion, it's kind of referring to 2 different things. And I want to add one more thing to this question is, if you want to use your own reference genome, like my lab really have a very nice assembling of this species, so you can always send your own reference genome to us as well, and we can help you evaluate if this is good to go or not.
Okay. A question from John Gamble. He's querying about the RNA-seq correlation plot. What parameter are you using to measure to create this plot?
So for the correlation plot, we are actually using the FPKM value for the analysis, as it's kind of just for us to visualize how my data distribute or how my sample cluster with each other. So we use the FPKM as input to create this plot. But if you have special request, like I want to use like the log-to-fold change, for example, the output or the results from the DSeq2 software, maybe we can also talk about this and customize the pipeline for you as well.
Okay, and a question from Alicia Vitello. She's asking, do we always perform RNA removal step? Is this for mRNA sequencing, or is it for which sequencing steps is this used?
So this one for RNAseq, we actually have multiple types of libraries, because for messenger RNA, they have the poly-A tail, which will be very easy for us to enrich them. So you can use poly-T oligo to enrich the poly-A tails or the messenger RNAs. So basically for messenger RNA, we will usually use the poly-A enrichment method to construct our library. But for some of the samples, such as FFP samples, you fix your samples in the parafilm and you store them for years. So those RNAs may degrade a lot or heavily. So for those messenger RNAs, it's very hard for us to use poly-A to enrich them. So in that way, we will use the ribosome removal library preparation. And for some other RNAs, like the long non-coding RNAs, like the circular RNAs or small RNAs, sorry, not small RNA, but for some RNAs that is not very efficient for us to use the poly-enrichment, we will use the ribosome RNA removal definitely. So for our link RNA and the circular RNA, we will definitely use the ribosome removal.
Well, thank you very much for those excellent answers, Wei. I think we're kind of coming to the end at the moment. If we haven't got through to your question, please, we will kind of get in touch through our local account manager to you. And if you have any questions post from this webinar, please feel free to get in touch. Thank you all for your time today. We hope you enjoyed the webinar and that you found it useful. We'll email you out a link to the webinar recording and to the slide deck, as well as a short survey that we'd really appreciate if you could complete for us. The survey will help ensure that we make future webinars even more relevant for you and your research goals. And in the meantime, please stay in touch. You can follow us on Twitter or on LinkedIn. And as you said, we can get in touch via the website at novogene.com. Thank you very much, and we look forward to seeing you all again at the next webinar.
Thank you.